Systematic reviews and meta-analysis provide Level 1 evidence. Secondary research provides a synthesis of the primary, individual research studies, which address the same specific and very focused question regarding a particular intervention. Pre-established rigid inclusion criteria are identified for the selection of studies. What is meta-analysis in research? Examination of data from a number of independent studies of the same subject to derive conclusions about that body of research. This analysis can increase the precision of estimates of treatment effects. Thus, a meta-analysis of randomized controlled trials is considered the most valuable when testing for causality. The content of this presentation is based on 4 sections: Introduction, Methodology, Data Description, and finally the software. What type of studies are we looking for to conduct a meta-analysis? Typically, but not necessarily, the meta-analysis is based on randomized controlled clinical trials. Non-randomized controlled studies can also be used. Case control studies is an acceptable alternative if there is no better evidence available. Review Manager is one of the most effective and free software to prepare and maintain Cochrane reviews. There are 2 versions of Cochrane Revman: Revman Web Online and Revman 5 Desktop. Revman Web is the online platform recommended for Cochrane intervention and flexible reviews. Revman 5 is the desktop version of the software used for other review formats, such as diagnostic methodology overviews, for non-Cochrane reviews, and for offline working. You can use Revman 5 alongside Revman Web if needed. You can download the desktop version for free after choosing your computer operating system. What data should you collect? You need a comprehensive information about each study, such as population and settings, for example, age, race, gender, socioeconomic details, disease status, duration, severity, comorbidities of the population, interventions and integrity of delivery, methods and potential sources of bias, outcomes, author's conclusions, citation, author contact details, sources of funding, etc. This information is required for references, description of included studies, risk of bias assessment, and sensitivity analysis.
So how can we minimize bias in data collection? So 2 authors should independently collect study characteristics and outcome data. This helps to reduce error and checks agreement on subjective judgments and interpretations. It is important to resolve disagreements, and this can usually be resolved by discussion or referring to a third senior author. You can try a pilot data collection process. This includes each person assisting and checking that criteria are consistently applied, and you may need to revise the form or instructions. Meta-analysis is characterized by a thorough and disciplined literature search. A clear definition of hypotheses to be investigated provides a framework for such an investigation. According to the PRISMA statement, an explicit statement of questions being addressed with reference to participants, interventions, comparisons, outcomes, and study design, PCOS, should be provided. It is important to obtain all relevant studies because loss of studies can lead to bias in the study. And abstracts are identified by a computerized literature search of electronic databases that can include PubMed, ScienceDirect, Sgourakis, ISI Web of Knowledge, Google Scholar, and Central, that is Cochrane Central Register of Controlled Trials. The PRISMA statement recommends that a full electronic search strategy for at least one major database is presented. Your trust or university librarian will help with the search to find potentially includable studies. Sometimes search reports thousands of studies for potential inclusion. This is a teamwork. Titles and abstracts first, you can exclude the vast majority of them after using your robust inclusion criteria. These are the basic steps in a meta-analysis. Firstly, you must identify comparisons to be made. Secondly, you identify outcomes to be reported and the statistics to be used. Then you must collect data from each relevant study and combine these results to obtain the summary of effect. You then can explore differences between the studies and interpret these results. But most importantly, you must publish it before someone else publishes the same subject. That we are conducting a hypothetical review as to the role of caffeine for daytime drowsiness. A reasonable comparison in our meta-analysis will be caffeinated versus decaffeinated coffee. Examples of outcomes are A, feeling asleep at the end of the trial, B, irritability, and C, presence of headache. Then we choose the effect measures of our proposed outcomes. For example, risk ratio for feeling asleep at the end of the trial, mean difference or standardized mean difference for irritability, and risk ratio for the presence of headache. To summarize, for each comparison, we select the outcomes, and for each outcome, we select the effect measure. All the aforementioned depend on the availability of studies or manageable authors' data. What kind of data can we include in our meta-analysis? Outcome data are of 5 common types. Dichotomous or binary data, where each individual's outcome is one of only 2 possible categorical responses. Continuous data, where each individual's outcome is a measurement of a numerical quantity. Ordinal data, including measurement scales, where each individual's outcome is one of several ordered categories or generated by scoring and summing categorical responses. Counts and rates, this is calculated from counting the number of events experienced by each individual. Time to event, typically survival, this is data that analyzes the time until an event occurs, but when not all individuals in the study experience the event, or censored data. Dichotomous or binary data, where each individual's outcome is one of only 2 possible categorical responses. Let's see the following example. We are analysing emotional exhaustion in medical trainees having a control group, a group that interventions A and B have been applied, and a third group where interventions A, B, and C have been applied. In our spreadsheet database, we need to extract each individual study data in a table as represented here. The uppercase N represents the total number of participants, and the lowercase n represents the number of participants presented with emotional exhaustion in each group before and after the intervention. Move on now to continuous data, where each individual's outcome is a measurement of a numerical quantity. Let's say that we are comparing the scores of emotional exhaustion amongst 206 junior physicians in the year 1997 with the respective scores of 518 junior physicians in the year 2007 after certain changes in their working conditions. We need to insert the relevant data of each study in our spreadsheet database as depicted in the table. We will need the mean value, the standard deviation, and the total number of participants. Other types of data are ordinal data, counts and rates, and time to event data, but let's focus on the dichotomous and continuous data at this stage, and you will be able to proceed to the former types of data when you're experienced enough in the latter. Dichotomous data expresses chance in terms of risk and odds. We will have the chance to discuss the effect measures for comparing groups with dichotomous data. Let's now move on to talking about risk ratio. In this example, we are analysing the impact of an intervention on the number of patients presenting with exacerbation of asthma. So the risk of the event with intervention is 1 out of 39. The risk of the event with control is 2 out of 40. The risk ratio is the intervention risk divided by control risk, which is 0.513. So how can we express this risk ratio in words? The risk of having a severe exacerbation of asthma with treatment was 51% of the risk in the control group. The intervention decreased the risk of asthma exacerbation by 49%. So where risk ratio equals 1, there is no difference between the two groups. When the 95% confidence interval intercepts through the 1.0 unit, this means that the risk ratio is unlikely to be statistically significant. Let's now talk about confidence intervals. So if a population is sampled 100 times, the means of the samples will lie within a normal distribution. 95 of these 100 sample means will lie between the shaded areas at the edges of the curve. This represents the 95% confidence interval. The 95% confidence interval can be viewed as the range within which one can be 95% confident that the true mean value lies. Let's now move on to a different example to explore odds ratio. So in this example, we are analysing the impact of an intervention in the number of patients presenting with exacerbation of asthma. So the odds of the event with the intervention is 1 out of 38. Odds of the event with the control is 2 out of 38. So in odds ratio, the intervention of the odds divided by the control odds, which is 0.5, equals to the odds ratio. So how can we express this in words? We can say the intervention halved the odds of asthma exacerbation, or the intervention decreased the odds to 50% of the odds in the control group. Where the odds ratio is 1, there is no difference between the two groups. And when the 95% confidence interval intercepts through 1, this means that the odds ratio is unlikely to be statistically significant. Now let's look at a different example to explore risk difference. So again, in this different study, we are analysing the impact of an intervention on the number of patients presenting with an exacerbation of asthma. So let's say that the risk of the event with the intervention is 1 out of 39, and risk of the event with the control is 2 out of 40. Well, the risk difference equals the intervention risk minus the control risk, which is minus 0.0244. How is this expressed in words? Well, intervention decreased the risk of severe exacerbation by 2 percentage points. Or you could say 5 out of 100 people experience an exacerbation in the control group, 2 less people experience exacerbation with treatment. Where the risk difference is 0, there is no difference between the groups. When the 95% confidence interval intercepts through 0, this means that the absolute risk reduction or absolute benefit increase is unlikely to be statistically significant. Continuing with exploring dichotomous data, let's now talk about choosing an effect measure. Let's now break down the effect measure of relative risk and odds ratio further. I'll give you 2 examples to make this information more palatable. So if a horse wins 2 out of every 5 races, its probability of winning is 2 out of 5, or 40%. In relative risk, the probability is the likelihood of an event in relation to all possible events. Now, if a horse wins 2 out of every 5 races, its odds of winning are 2 to 3, expressed as 2 to 3 ratio. Odds compare events with non-events. We use relative risk as a ratio of probabilities. For example, myocardial infarctions in those who smoke cigarettes compared with those who do not, while the odds ratio is used in cases of presence to absence of an exposure. We already know about a specific outcome to presence to absence ratio of cigarette smoking in those who had an MI compared with the same ratio in those who did not have an MI. It is only appropriate, therefore, to use relative risk for a prospective cohort study. Odds ratio can be used to describe the results of a case control as well as prospective cohort studies. We will now have a look at the way we transform data for dichotomous outcomes. Some authors, instead of presenting their data using 4x4 tables, give odds ratios or risk ratios. For this, we need to convert the data of our included studies into risk ratios or odd ratios in order to conduct our meta-analysis. On this site, you can find a practical meta-analysis calculator which can transform your data. Let's now have a look at the way we can transform data for continuous outcomes. The standardized mean difference effect size, D, is designed for contrasting two groups on a continuous dependent variable. The standardized mean difference is the difference in mean outcome between groups divided by the standard deviation of outcomes amongst participants. The standardized mean difference can be computed from means and standard deviations, a t-test, or a one-way ANOVA, depending on the way several included studies in our meta-analysis present their outcomes. Some authors prefer to present the data using mean, standard deviation, and number of participants. This layout provides data from a t-test or a one-way ANOVA. You can use this calculator to transform collected continuous data to a standardized mean difference for each of the included studies. In this next section, we're going to be talking about working with the Cochrane Collaboration Review Manager, the RevMan meta-analysis software, which you need to be familiar with. This is the interface of the program that welcomes you to the review wizard. Clicking Next brings you to this screen, where you can now choose the type of review that you would like to conduct. Let's say that you want to conduct an intervention review. You can also choose the other options such as diagnostics test accuracy review, methodology review, overview of review, and flexible review. You can choose the title of your review. The program will guide you through this process. Options include intervention for health problem, or intervention A versus intervention B, or intervention for health problem in a participant group or location. In this example, we've chosen the second option. So that is a minimally invasive video-assisted thyroidectomy versus conventional thyroidectomy for thyroid nodular disease or a low-risk papillary carcinoma. You then come onto this screen of which stage should the review start in, and most of the time we want a full review to put everything into place. You then come onto this screen where at the top you have the menu buttons. On the left-hand upper panel you have the sections of your meta-analysis to fill in, and below that you have the information that's needed for your statistical analysis, and as well as the visual presentation of the outcomes. The right panel presents all the information filled in each of those sections on the left-hand panel. stage is to go through the Prisma flow diagram to identify studies that you would like to include in your meta-analysis. You then define your comparisons, the outcomes in each comparison, and the effect measure according to the type of data. At the end, you fill out the included studies data to your spreadsheet. The section Reference to Studies is expanded when you use the right or left mouse click on the included studies. The software responds with the new study wizard, and it will guide you through the process of adding one of your studies that you've chosen to include. Repeating the same process, you'll add all your studies that you want to analyze in your meta-analysis. And most of the time, the leading author and the year of the study is all that's needed in this stage. After completing the process of adding the studies that you would like to include in your meta-analysis, you'll see that they're all present there on the left-hand panel under Included Studies. On the right-hand panel, you can optionally add a few more important bullet points, but most importantly, you need to use your critical judgment and define the level of risk in each of the elements that might be contributing to bias in the individual study. The next step is to define our comparisons. So you right-click on the Data and Analyses section, and the program responds with a new comparison wizard. And in this hypothetical example, we type short-term outcomes and then click Next. As we have mentioned already, so after defining your comparisons, you then need to add outcomes for each of those comparators that you've selected. So if you prefer, you can add another comparison, for example, long-term outcomes, and add the outcomes in each comparison at a later stage. Let's say that we are now adding outcomes for one of our comparators, which is short-term outcomes. We need to firstly choose the type of data that will be used for the analysis of that particular outcome, and in this example we've chosen continuous. And then you will see a description of the information that needs to be filled on the right hand side of the wizard according to the type of data that you choose. Occasionally you might find that not all authors of the included studies provide their data in the form of the mean, standard deviation, and number of participants for continuous outcomes. Then we need to click the option of generic inverse variance, but we then need to transform all included studies data with using a meta-analysis calculator. When looking at short-term outcomes, we can now add one of our first comparators, which is operative time. You then add the groups that you are comparing, which in this example was minimally invasive video-assisted thyroidectomy group versus the conventional surgery group, as shown in this example. We then click Next. The software will then proceed to give us the short-term outcomes in the left-hand column under Data and Analyses. And on the right panel, you will see that the operative time has been presented in a table. It is a continuous outcome, therefore we'll need the mean values, standard deviation, and the number of participants in each group. Following the same steps that we have already shown you, we've then created our second comparison, which is long-term outcomes. One of the long-term outcomes that we've looked at in this case is transient palsy, which is shown in the right-hand panel. This is a dichotomous outcome, therefore the table to fill the data is different from the operative time table, which uses continuous data. If we expand the Data Analysis tab on the left, we can see all of our comparators and the outcomes under each comparison, which could be more than one. A lot of the time you might find that all of the included studies don't necessarily provide data for each of the included outcomes. To include only the relevant studies to your chosen outcome, you first right-click on that outcome you're going to analyze, and the program responds with a new study data wizard. All the studies that you have added under the heading of included studies will be shown. So holding the control key down, you choose which of the studies provide analyzable data according to the tables in your spreadsheet database. Then you click on the Finish button. In this example, the operative time outcome is chosen on the left-hand panel, and we have included all the studies to be used for this outcome, as we showed you in the slide before. It's highlighted on the right panel as well, and a table is ready to fill with the data from your database. We now have to transfer all of our data that we have collected from Excel into the database formed on the software. You might have to do this manually, as copy and pasting might be tricky. The RevMan software will automatically provide the mean difference and the confidence intervals of each individual study, and the same parameters for the outcome under analysis. What do you think? Is there any detected statistically significant difference here? The difference is yes, since the confidence interval is not intersecting 0. The p-value and the test value is given by the program, as shown under the heading of Test for Overall Effect. We repeat the same new study data wizard for all of our outcomes in the way that we've shown in the previous slides. In this example, we have chosen all the studies as all the included studies provide analyzable data for the outcome transient palsy. Having gone through this presentation, you should now be able to define the overall effect, which in this case is in the form of odds ratio and confidence intervals, and comment on the statistical significance of this outcome. You will notice that 0 patients had transient palsy in 2 of the included studies. So it's advisable to replace 0 with a very small number like 0.1 or 0.5 for your analysis. That concludes our first part to our guide on performing meta-analyses. In our next video, we will discuss heterogeneity among studies and the use of the fixed term versus the random effect tests, as well as visually presenting your data in the form of forest plots, funnel plots, and the analysis of the risk of bias. Thank you for listening, and we look forward to hearing your ideas.